Good day, everyone. I am delighted to welcome you to African Bioimaging Consortium, a microscopic training initiative. I am Dr. Ibrahim Abdullahi from the Faculty of Veterinary Medicine, Department of Veterinary Anatomy, University of Abuja, the Federal Capital Territory, Nigeria. I'm going to take you through the first lecture in the introductory series in microscopy training with the topic Introduction to Microscopy, our learning objectives. At the end of this lecture, you should be able to explain between the terms microscope and microscopy. You should be able to discuss the principles behind the light microscopy. You should also tell the different types of microscope, understand where the focal plane is. You should be able to explain how refractive index matching improves results, explain the differences between resolution and magnification, and lastly, you should be able to explain how objective lenses work. What are microscopes? The term microscope originates from two Greek words, micros and scopio. Micros is a root word for microscope, microscopy, microorganism, microbiology, microanatomy, microenvironment, and many other scientific terms that deals with very small things or objects. Literally, micros stand for small or tiny, and scopio means to view or to look at. When you combine the two words, it gives us a term microscope, which is a physical instrument that is used to enlarge the size of an object, making small specimen visible. What do you do with it? With the microscope, you can be able to observe a very small things or objects that is invisible or hard to see with the naked eye. For example, looking at the things we cannot see with the eye, with the naked eye, like the household dust, the bread mold, the fly eye, cat flea, the cancer cells, under the microscope appear bigger and provide sufficient details for image analysis. With the microscope, you can be able to magnify objects that reveal some interesting microstructures that help you to study more details about that specific object. For example, looking at the sand under the microscope, where individual grains appear like a small stone, each with different colors and shape. We study biology across a wide range of scales and sizes. It is important to understand what size each type of microscope can handle for several practical and scientific reasons. This will enable a researcher to choose the right tool for his observations. And it also helps us to understand the complexity of biological structure that occurs at different scales. For example, looking at the unaided eye, it can see objects with size ranges from 100 micrometer to about 10 meter and above. Objects like the chicken egg, frog egg, length of some nerve and muscle cells, human height. The light microscope can see objects with size ranges from 100 micrometer to about 0.2 micrometer. Things like eukaryotic cells, nucleus, most bacteria, and mitochondria can all be seen with the light microscope, while the electron microscope can see objects with size ranges from 0.2 micrometer to about 0.1 nanometer. Understanding this phenomenon is very important, and it helps a researcher to have a proper research planning, accurate analysis, efficient use of time, and of course, it helps him to have a clear scientific findings. Where did it all start? Long ago, Greek thinkers were extremely interested in seeing and understanding the natural world, though they don't have the microscope, but they imagined tools that could help them see the unseen. That is how the idea of seeing the small started. In 1284, Salvino di Amati, who is an Italian from Florence, was credited as the first person to create wearable eyeglasses. Salvino did not work with a microscope directly, but his works with optic lenses for helping vision lay the foundation for lens use in science. Roger Bacon, who is an English philosopher known for his work in science, optics, and experimental method, he studied and wrote about lenses, magnification, and the science of light and vision. If you look at it, both Salvino di Amate and Roger Bacon were not the inventors of the microscope, but they play a key role in early use and understanding of lenses. In 1590, the first actual microscope was invented by an eyeglass maker, the Charles Jensen of Netherlands, along with his father, Hans Jensen. In olden days, there were no factories making glasses, so skilled people like the Charles Jensen and his father hand-made lenses and put them into frames for people with poor vision. While working with lenses for eyeglasses, they experimented by placing two lenses at the ends of a tube. That was the first device capable of magnifying tiny things more than a single lens. Uh, in 1609, Galileo Galilei, who is an Italian scientist known for his work in astronomy, physics, and inventions, he modified the compound microscope, though he is not the inventor of the microscope. In similar developments, in 1625, Giovanni Faber, who is a German-born physician, a botanist, and a member of Accademia di Lincei, which is an early group of scientists in Italy, was the first person to use the word microscope. Giovanni was inspired by the word telescope, which had already been coined earlier in 1609 for the instrument used to view a distant object. He wanted a similar word for the instrument used to view at tiny objects, hence the name microscope. In 1665, an English scientist, Robert Hooke, helped to establish the microscope as a vulnerable instrument in scientific inquiry. In his book, Micrographia, Robert Hooke coined the term cell after observing a thin slice of cork under the microscope. Robert Hooke showed that microscopic observations could reveal the hidden structure of an object. Around 17th century, a breakthrough by a fabric merchant, Anthony Ballion, took the microscope to the next level. He made a very powerful single lens microscope with magnification of up to 200 to 300 times, which is far superior to many compound microscopes of his time. But Leeuwenhoek was the first to observe and describe living microorganisms, which he called animals. To the modern microscope of today, which evolves into several types with advanced technology and AI incorporations. In summary, microscopes have changed how we understood the world starting from 1600. Why is this lengthy history important? It is important because one, it shows the progress of human curiosity to know about the nature. Two, it helps us to appreciate the tool, which is not just a lab instrument, but rather a result of centuries of curiosity and inventions. Three, it helps us to understand how foundation for modern science was made, because without this, many branches of science like medicine, pathology, microbiology, histology would not exist. Microscopy. What is microscopy? We learned that microscope is a physical instrument, is a hardware, is a gadget that is used to enlarge the appearance of an object, making small specimen visible. While microscopy is a science, it is a discipline. Microscopy is everything involves in studying tiny things, not just viewing it, but learning how, why, and what are you seeing. Microscope must accomplish three essential tasks to be effective in biological and scientific observations. These tasks include magnification, resolution, and contrast. In magnification, what it does is that it enlarges the appearance of an object, making small specimen visible. This is important because it allows observation of small structures, and this is essential for detailed study. Resolution uh, is a microscopic ability to distinguish between uh, similar looking structures. It's a microscopic ability to distinguish between closely spaced points as separate. A microscope with uh, uh, higher magnification but poor resolution will show unclear or merged details. So resolution is critical for distinguishing between similar looking structures. Contrast. Contrast, it is the ability to see differences in color intensity between different parts of a specimen and its background. This is important because biological specimens are often transparent. That is, without contrast, they appear unclear or invisible even under the microscope. In simple terms, contrast is a key to visibility in microscopy. That is why stains, contrasts, or other specialized techniques are employed to enhance contrast. In summary, this task, magnification, resolution, and contrast, are essential for accurate, meaningful, and detailed microscopic observations. Understanding parts of a microscope can be made easier by categorizing them into four distinct groups. One, those parts that provide support and stability. Two, the optical components. Three, the illumination system. And four, the focusing system. Starting with the first part, those parts that provide support and stability forms the structural framework of a microscope. This uh, includes the base, the arm, and the body tube. The base is usually found at the bottom. It houses the light source and the switch. Arm is a part that connects the body to the head of a microscope, and it is used in carrying the microscope. The body tube holds and aligns the two sets of lenses, that is, the ocular lenses and the objective lenses. The optical components are those parts that are responsible for magnifying the image and bringing it into focus. This includes the ocular lenses, the resolving nose piece, and objective lenses. Starting with the objective lenses, these are primary magnifying lenses. In fact, without these lenses, a microscope is just like an empty tube. So they are very critical and composed of four sets of lenses. The times four objective lenses, otherwise called scanning objective lenses, times 10 objective lenses, also called low power objective lenses, times 10 times 40 objective lenses, also called the high power objective lenses, and times 100 objective lenses, which is globally recognized as the oil immersion. The resolving nose is a part of a microscope that allows an observer to change between different objective lenses. Ocular lenses are secondary magnifying lenses, and these lenses are the ones closest to the observer. Usually, they come in times 10 and times 15. So care should be given in knowing which number of the ocular lenses are you dealing with. This will help to arrive at a correct magnification. The illumination system are those parts that control the amount of light entry reaching the specimens. These include the light source, the condenser, and the iris. The light source transmits light from below onto the specimens, and the iris controls the amount of light entry through the condenser, and the condenser helps to focus light onto the specimens. The focusing system, that is the last part, are those parts that are responsible for bringing the specimens into clear details. These include the coarse adjustment knobs, the fine adjustment knobs, the stage clip, and stage control. The coarse adjustment knob is that part of a microscope that is used to quickly bring the specimens for rough focusing. It usually lifts the stage up and down. The fine adjustment knob is a part of a microscope that quickly sharpens the image for clear observations. And the stage is a flat surface where you place your slide. The stage clip is a part that helps to scale the slide in place. And lastly, the stage control is responsible for moving the slide to and fro, left and right. Overusing coarse adjustment knob on higher power will crack the slide. So it is important to know these parts of a microscope for correct usage and prevention of damage. How do we see under the microscope? Cells are too small, too clear, and too uniform. This is because animal cells lack a natural pigment, unlike plant cells that have chlorophyll. They are extremely thin, making them light permeable and hard to see under the microscope without enhancement like stain, special technique, or contrast. What is contrast? Contrast is what makes details visible. Contrast is a key to visibility in microscopy. The following six histological sections, level 3A to F, are stained and viewed under different contrast techniques, highlighting how staining and contrast reveal specific structures in tissues. These are another four histological sections, stained under different techniques, where structures stand out against their background, aiding visibility and interpretation. What are the types of microscope? Microscope, basically, microscope can be classified on the basis of one, uh, radiation type, and two, type of image formation. Understanding different types of microscope is important because Each type of microscope has specific strengths, applications, and limitations. On the basis of radiation type, we have light microscope and electron microscope. On the basis of radiation type, we have Zeiss microscope producing two-dimensional images and Zeiss microscope producing three-dimensional images. What are light microscopes? Light microscope is a type of microscope that uses a visible light as their source of illumination. These are the most commonly used type of microscope. All the types of light microscope virtually produces two-dimensional images with some few exceptions. Types of light microscope. Light microscope can be divided into bright field microscope where uh, the light passes through the specimens and the specimen is always against the light background, hence the name bright field microscope. Another type of light microscope is a dark field microscope. In this type of microscope, the light hits the specimen from the side and the specimen is always against the dark background. This type of microscope is ideal for observing live specimens. First contrast microscope is a kind of microscope that enhances contrast directly onto the specimens. And this type of microscope is ideal for observing live on-stain specimens. Fluorescence microscope is another type of light microscope that highlights specific structures with high specificity and contrast. For example, uh, the nucleus is stained blue, clearly seen against the green cytoplasm. So this type of microscope stains specific parts with high specificity and contrast. Dissecting microscope is another type of light microscope with unique features. Part of the features of this microscope are: 1. The light source is found at the objective lenses, and the stage is usually found at the base. Unlike in other four types of microscope we have just explained, the light source is at the bottom and the stage is at the middle. So in all the four types of microscope, the light travel, uh, the light is transmitted from below onto the specimens. But in dissecting microscope, the light travels down, hits the specimen, and bounces up to the objective lenses. Another unique feature of this type of microscope is that uh, it has a very poor magnification of about 7.5 to 40 times. In contrast to the other four types of microscope, which have a magnification of about 40 times to 1000 and above. This dissecting microscope is used to view a very large specimen. In fact, the whole organism can be observed under this type of microscope. It gives the observer the ability to manipulate the specimens, thereby dissecting the specimens while looking through the microscope. This type of microscope does not require the glass slide. It does not require the specimen to be placed on the glass slide. In contrast to all the four types of microscope that requires the glass slide, for the placement of the sample. Electron microscope is a type of microscope that uses electron beam as a source of illumination. Basically, we have two types of electron microscope: scanning electron microscope and transmission electron microscope. Starting with the scanning electron microscope, as the name suggests, scanning, it examines the surface morphology of a specimen. So this type of microscope displays three-dimensional black and white images of a thin section of a cell, highly detailed, illustrating the surface morphology, while the transmission electron microscope displays a two-dimensional black and white images of a thin section of a cell, clearly showing internal details. This is because light passes through the specimens, revealing ultrastructural details. How lenses bend and focus light to form images. In this context, it is important to differentiate between the terms refraction and refractive index. Refraction is the bending of light as it moves between different materials. This occurs because light changes speed when it moves between different media. Every material has its own power for bending light, and this is called refractive index. The common materials that are used in microscopy are air, oil emulsion, glasses, and many more. So each of these substances have different uh, capacity to bend light, and that is called refractive index. In microscopy, when we are looking at the tiny object under the microscope, we want the image to appear as clear and sharper as possible. But when light bends too much, it causes the image to appear unclear. Refractive index matching means using substances with similar refractive index. This gives a much clearer details of an object. If you look at these diagrams illustrating uh, two uh, aspects. One, viewing a particular specimen without oil immersion, and the second is viewing a particular specimen with oil immersion. Usually, air has a low refractive index when compared to glasses. So when you use air gap under times 100 objective lenses, it causes light to move away from the objective lenses, as you can see on the diagram. If you use air gap under times 100 objective lenses, you lose light and clarity because the two substances has different refractive index. On the other hand, uh, with oil immersion, if you look at it, when observing a substance, when you use materials with similar refractive index, it allows the light to move into the objective lenses, and this brings a very clear and sharper image. So when you use substances with similar refractive index, there is no loss of light, and the images will appear very clear with high quality. Oil immersion has a refractive index of about 1.515, and the glass has a refractive index of about 1.50. So if you look at it, the two substances have a similar refractive index. Unlike, on the other hand, the glass slide has a refractive index of about 1.5 and air has a refractive index of about 1.0. So the gap is too wide between the two substances. So always use substances with a similar refractive index. And that is what is called refractive index matching. Using substances with similar refractive index. That gives a better quality images. Now focus and the focal plane. In this context, it is very important to understand between the terms focus, focal plane, the focal point, and focal length. Focus is action. It is a process of making an object or an image to appear very clear. The focal plane or the focal point first is a particular spot where light rays converges at a point after passing through the lenses. The focal length is a distance from the focal point to the center of the lenses. Short focal length signifies the lenses strongly uh, uh, bends light, and the long focal length signifies the lens uh, bends light a little. In microscopy, we want the image to appear as clear and sharper as possible. So if we use a lenses with shorter focal length, meaning the images will appear, the lights, the focal length will bend light too strongly and the images will form too quickly. Focal plane. A focal plane is defined as an imaginary flat surface. A focal plane is an imaginary line that passes through the focal point. When substances or when a sample passes onto the focal plane, the images appear clear with fine details. But when samples fall below the focal plane or above the focal plane, objective lenses. As we mentioned earlier, these are the primary magnifying lenses. Without these lenses, a microscope is just like an empty tube. So there are notable markings on the objective lenses, which we are going to study in details in the subsequent lectures. But the basic markings here are the magnification, the numerical aperture, the tube length, and the cover slip thickness. If you look at the four different objective lenses, we have, it is clearly written on them with different magnification. The oil immersion has a magnification of 100 times, meaning it enlarges a particular specimen 100 times than its original size. The higher power magnifies a particular specimen 40 times than its original size. So also the low power magnifies image 10 times its original size. The scanning magnifies the image 4 times its original size. So it is clearly written, the magnification is written on the respective four types of the objective lenses. The next one is the numerical aperture, which differs across the objective lenses. The higher the numerical aperture, the better resolution and clear images. So on the oil immersion, the numerical aperture is 1.25. On high power, it is 0.65, and on low power, it is 0.25. And what happens if I try to image through the wrong thickness? Yes, a number of things will happen. Among them is you are going to have a focus problem, a poor contrast, and of course, you are going to have unclear images. Resolution and magnification. Resolution is a microscopic ability to distinguish between two similar looking structures, or, on the other way, it is a microscopic ability to distinguish between two closely spaced points as separate, while magnification is the ability of a microscope to enlarge the appearance of a tiny object that is invisible or hard to see with naked eye. So in summary, magnification is a process of enlarging the appearance of an object, while resolution is a process of making that particular object to appear in details. As you can see here in the diagram, uh, uh, this is clearly magnified but poorly resolved. On the other way, the three dotted points are clearly enlarged and clearly seen. In conclusion, microscopy has bridged the gap between visible and invisible. It is about mastering magnification, resolution, and contrast. Microscopy has changed science and will continue to drive discoveries in the future. Thank you for listening.